In the previous parts of this lecture, we saw that the energy-rich substrates are oxidized via glycolysis followed by Krebs cycle to release finally from NADH and FADH2 and some ATP. The electron transport system or electron transport chain is the one that ultimately releases the energy that is trapped in the following molecules, that is NADH and FADH2. This energy is then utilized for synthesizing ATPs. NADH and FADH2 are oxidized through what is known as electron transport system to release their energy. In the process of electron transport system, electron from NADH and FADH2 pass through one carrier to another and are ultimately passed on to oxygen, resulting in formation of water, H2O. This electron transport chain is located inside the inner mitochondrial membrane. It's not difficult to understand the electron transport chain if we understand the following facts. That one hydrogen atom is actually just one electron and one proton. Therefore, H plus ion is only a single proton. So, a hydrogen atom becomes hydrogen ion, only single proton, after it gives away its only electron. Also, oxidation, we call oxidation when there is addition of oxygen to an atom or a molecule, or removal of hydrogen, or removal of H ion, or removal of electrons. And reduction is gaining hydrogen or removing oxygen or receiving electrons. You can remember that if you receive something in negative, you are actually getting reduced. The H atom of NADH, that is one proton with its electron, is passed on to complex 1, which keeps the electron and discards the proton, that is H plus, in the intermembrane space. The elements of electron transport chain, as they receive the electrons, they pick up one H ion from the matrix while accepting this electron from the previous member. As they pass the electron to the next element, this H ion or proton is released into the intermembrane space again. The next element repeats the same thing. So the electrons are passed on with corresponding shift of proton from matrix to the intermembrane space till the electrons are finally accepted by oxygen. And this procedure leads to high concentration of H ions in the intermitochondrial membrane space. So a concentration gradient is created across the inner mitochondrial membrane. That is a high concentration of H ions in the intermembrane space and a low concentration of H ions inside the matrix. So these H ions move back into the matrix via ATP synthetase. Energy released during this entry is utilized by ATP synthetase to synthesize ATPs. This is a story which we will explain further by using animation. Some more facts for simplification of electron transport system is NADH dehydrogenase, that is an enzyme which removes H from NADH, is known as complex 1. FADH2, which was generated during Krebs cycle, is also known as succinate dehydrogenase and also known as complex 2. Complex 3 is cytochrome BC1 complex, and complex 4 is cytochrome C oxidase, and cytochrome C oxidase or complex 4 consists of 4 parts: cytochrome A, cytochrome A3 and 2 copper centers. Complex 5 is ATP synthetase. What is oxidative phosphorylation? Is that ADP is synthesized to ATP using the energy released by oxidation, which is removal of electrons. So the first step, electrons from NADH are oxidized by complex 1, that is NADH dehydrogenase. That is, it accepts the H atom from NAD via complex 1, ubiquinone, and then ubiquinone also accepts the H atom from FADH2. So ubiquinone receives H atoms from both NADH via complex 1 and directly from FADH2. So NADH gave its electrons to complex 1, which gave it to ubiquinone, and FADH2 gives it directly to ubiquinone. Remember, FADH2 was generated during oxidation of succinate in the citic acid cycle. On the right side you can see succeeding acid forming malic acid. FADH2 is actually an enzyme, also known as succinate dehydrogenase or complex 2, and it converts succinate first to fumarate while taking H from it and finally to malic acid. So one more step in Krebs cycle is mentioned here. The ubiquinone, which had accepted electrons from complex 1 and FADH2, is reduced to ubiquinol because it picks up an H ion also from the matrix. So it ultimately has an H atom. It is reduced to ubiquinol. The ubiquinol then gets reoxidized back to become ubiquinone by transferring the electrons to cytochrome C and releasing the H ion into the intermembranous space. Remember, this H ion was picked up from the matrix. Ubiquinone ultimately sends electrons to cytochrome C via cytochrome BC1 complex or complex 3. So ubiquinone gives to complex 3, which ultimately gives it to cytochrome C oxidase. So both NADH and FADH2 give their electrons to ubiquinone, who then sends them to cytochrome C via complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane of mitochondria. Cytochrome C is a mobile carrier. It transfers electrons between complex 3 and complex 4. As we saw before, complex 4 is cytochrome C oxidase. It contains cytochromes A and A3 and 2 copper centers. So we can see that cytochrome C accepts the electrons and then goes for self-oxidation to cytochrome C oxidase. As complex 4 receives electrons from cytochrome C, again it picks up H ion from the matrix of the mitochondria. So the scheme of electron transport is from NADH and FADH2 to ubiquinone via complex 1 in case of NADH to cytochrome BC1 complex or complex 3 to cytochrome C, which finally goes to cytochrome C oxidase complex 4. Finally, the electrons are accepted by oxygen from complex 4. Complex 4 finally passes the 2 electrons to oxygen atom and once again releases the H ion into the intermembranous space. The oxygen atom, after receiving 2 electrons, picks up 2 protons from the matrix itself and completes 2 H atom to form H2O, which moves out. And this further reduces the concentration of H ions in the matrix. This animation will make electron transport chain absolutely clear. The inner membrane of mitochondria is shown to be light blue in color and the outer membrane of mitochondria is shown to be dark blue in color. The space between the inner and the outer membranes is the intermembranous space and the space inside the inner membrane of mitochondria is the mitochondrial matrix. We can see some elements of electron transport system located in the inner membrane of mitochondria. The matrix of the mitochondria has some H atoms, H ions, NADH where H is shown to be 1 proton and 1 electron, FADH2 where again the 2 H atoms are shown to be 2 protons and 2 electrons. NAD gives its H atom to complex 1 which keeps the electrons and the H ion is thrown in the intermembranous space. FADH2 gives its electrons to complex 2. H ions are thrown in the intermembranous space. The electrons from complex 1 and complex 2 go to ubiquinone, which accepts H ions to become ubiquinol. Electrons are transferred to complex 3 and H ions they go in the intermembrane space. The electrons then go to cytochrome C from complex 3 and H ions are again similarly moved into the intermembrane space. Cytochrome C transfers the electron to complex 4. Complex 4 gives the electrons finally to oxygen atoms. which takes more protons from the matrix, water is formed which moves away. So we can see that H ions, very low concentration in the matrix and a high concentration in the intermembranous space. So revision again, NADH electron transfer sequences from NADH to complex 1, which gives to ubiquinone or coenzyme Q it is also called. H ions into the intermembranous space, FADH2 gives to complex 2 and H ions go to intermembranous space. Ubiquinone gives to complex 3 which gives H ions again go out which gives to cytochrome C. Cytochrome C finally gives electrons to complex 4 which gives the electrons to oxygen. H ion also go out again. Oxygen takes electrons as well as protons from the matrix, forms water and water moves out. Fine, but how are ATPs produced? We haven't seen them till now. ATPs are produced via a process called chemiosmosis. We have seen that during electron transport chain, H ions get concentrated in the intermembranous space of mitochondria while the concentration of H ions is reduced in the matrix. So, H ions move from a high concentration area, that is intermembrane space, into the lesser concentration area, that is into the matrix, via ATP synthetase, which forms a channel. As H ions move into the matrix through ATP synthetase, ATPs are synthesized, which are released into the matrix. You may ask a question, why then we focus on electron transport system, while it is the H ions which are actually running the show? Chemiosmosis is a process, it occurs in photosynthesis also, which requires a semi-permeable membrane, which is the inner mitochondrial membrane in this case. It requires energy for proton pump, which we will see, and a proton gradient resulting from pumping of protons, which is a high concentration of H ions in the intermembrane space and low concentration in the matrix of the mitochondria. And fourth requirement is ATPase or ATP synthetase. The energy, that is the point number 2, for pumping the protons out comes from the high energy electrons which release energy each time they are transported to a lower energy stage, that is between the electron transport system. As they are processed, they keep releasing energy after every transfer. Here we can see in the whole mitochondria a high concentration of H ions in the between outer membrane and inner membrane of the mitochondria, the ETS located in the inner mitochondrial membrane, a low concentration of H ions in the mitochondrial matrix. As the H ions move from high concentration into the matrix, it has to occur through ATP synthetase, which forms a channel, and as H ions enter, ADP is converted to ATP by ATP synthetase.